This is TP53 gene. TP53 is located within our DNA, which has a very important role in translating a type of tumor suppressor protein called p53. They have the ability to regulate cell cycle, which indirectly aids in the repair of damaged DNA and inducing apoptosis. In a normal cell cycle, the state of p53 proteins are highly unstable, and that is why they exist in very low amounts within a cell. Only when a particular lesion is integrated into the DNA will then p53 proteins is stabilized and produced at a larger amount. With all that being said, they are now able to bind and activate the expression of a promoter gene called WAF1. This interaction leads to the production of another important protein called p21. In order to showcase the importance of p21 in the cycle, let us explain briefly how the cell cycle actually operates. There are 4 important checkpoints within the cell cycle. The G1, also called the gap phase, is where the cell starts to grow and prepare for DNA synthesis. The next station is the synthesis phase, or the S phase. It's where the cell actually starts to synthesize its own DNA. And for a cell to progress from G1 to S phase, an enzyme called cyclin-dependent kinase, or CDK, must first bind to one or more G1 cycling proteins in order to do its job. What happens next? Cyclin-CDK complex can then phosphorylate key proteins needed for the cell to enter the S phase for DNA synthesis. Now let us come back to the role of p21 proteins. In this event, p21 protein has the ability to bind to activated cyclin-CDK complex and blocks its enzymatic ability. When this happens, cyclin-CDK can no longer phosphorylate key proteins and therefore preventing the cell to go into the S phase and stays in G1. If a cell rests in G1 phase for too long, this cell will undergo apoptosis. Ow! Up to this point, we are able to appreciate and understand that the importance of p53 proteins in regulating the proliferation of tumor cell is crucial. Unfortunately, mutation of TP53 in cancer patients is highly common. When this happens, TP53 is inactivated, and the increased production of p53 proteins in the presence of damaged DNA ceases to exist. This then leads to an inactivation of WAF1 gene, and thus inhibit the production of p21 proteins. Not only that, but this also means that the enzymatic activity of cyclin-CDK complex will continually be activated, allowing the cell to proceed into the S phase to complete the cell cycle along with the damaged DNA. As you can imagine, this progression of uncontrolled cell proliferation with damaged DNA will eventually accumulate unwanted group of cells that have the ability to become cancerous.